Okay, hi everyone. Welcome to this Metabolomic Society podcast. This is the sixth in the series. They've been very helpful and successful in the past, so hopefully they will be today. This podcast, we're going to focus on quality assurance and quality control in metabolomics, which is a really important area, often overlooked, seen as something extra to do, but it's something that is really, really important. Hopefully the next 55 minutes, you'll understand why it's important, how it's applied, and how you can apply it in your laboratory as well as how we should report it in our scientific publications, presentations, etc. So I'm going to go around and ask the expert panelists to introduce themselves in a minute. I'm just going to introduce myself. I'm Professor Warwick Dunn. I work at the University of Liverpool in the UK. And I've had a long interest in quality assurance and quality control from many, many years ago, discussing it with Ian Wilson, who's one of our expert panelists today, including with the development and ongoing great work that MQAC, which is the Metabolomics Quality Assurance and Quality Control Consortium, which you'll hear about hopefully during the podcast as well. And my co-chair today is Dr. Michael Whittin, over from Germany. So I'm just going to stop the slide presentation and then go around and ask the panelists to introduce themselves. So I'm going to go as it's set up on my Zoom. So Annie, would you like to introduce yourself?
Yeah, hi, my name is Annie Evans. I'm the head of research and development at Metabolon, covering our untargeted as well as our targeted workflows for both lipidomic and small molecule. My real passion for quality control and quality assurance is towards clinical work. I feel, and I hope everybody who hears this podcast is in so much belief that metabolomics has the power to change human health care. And to be able to get there, we have to have extreme quality assurance and quality control. And so that's where my passion lies in this space.
Okay, thank you, Annie. I'll be a gentleman and Jennifer next.
Hello, everybody. I'm Jennifer Kerwin. I lead the metabolomics platform at the Berlin Institute of Health, which is part of the Charité Hospital in Berlin, in Germany. I came into metabolomics from a veterinary angle, and I work a lot in precision medicine. And for the same reason as Annie, I think that quality assurance and quality control is vital for translation, but also for robustness of results. And I should also mention that I'm the current committee chair of the Metabolomics Quality Assurance and Quality Control Consortium, of which there are several members talking today.
Jonathan next.
Hey, Rick. So I'm Jonathan Mosley. I am a research chemist at the Environmental Protection Agency in the United States. I should say that everything that I'm going to say is my own opinion and not that of the EPA. However, the EPA is what brought me to metabolomics. And so a lot of my passion surrounding quality assurance, quality control, is for metabolomics applications in regulatory toxicology and sort of ecological assessments as well. But also, like Jennifer, I am a member of the coordinating committee of MQAC, and I've actually been involved with a few others on the call with MQAC since its inception. And so a lot of my perspective has been shaped by my involvement in that consortium as well.
Brilliant, thank you. And finally, I've worked out the technology. Ian, if you'd like to introduce yourself.
Okay, I'm Ian Wilson, and I guess I'm a dinosaur from the last century because I've been doing metabolomics since before it had a name. I'm currently a visiting professor at Liverpool University and Imperial College. Yeah, I mean, Rick mentioned talking about QA and QC a long time ago, it seems now, in the Husamet project, and it's impressive to see how it's grown since then. And I hope that I have some things that are interesting for you to hear today.
Brilliant. Thank you, Ian. So just so everybody's aware, we are recording this. It will be available on the Metabolomics Society website in the coming weeks and on YouTube. We now have a YouTube channel. For any of the non-panelists, if you have questions, then feel free to put them into the chat. Please make sure you keep muted throughout the podcast, but please enter questions into the chat. And if we have time at the end of the podcast, we'll get into those and answer some of those questions as well. So please feel free to interact in that way. Okay, so let's start off and think about, if I can find them. The questions. Okay, so the first question that we've been asked, and I'm going to ask Annie to answer this, is the simple question of what is QA and what is QC, and how do these differ?
Yeah, really important. So I always like to think of this, and I'll give some examples because I often think examples is the best concrete way to really understand the difference. QA is all the things that you do to build around to ensure you have the ability to collect high quality data. So examples of this would be creating SOPs for your lab. Doesn't have anything to do with actually executing, run the experimental samples, but is in establishing the process that says this is a quality process that you can follow to ensure that you're going to get good quality data. Other examples of this are competency testing and proficiency testing for your lab staff. Yes, your staff may have an SOP, but do they know how to follow it? Have they demonstrated that they can do it? Again, that is part of QA to say you have the ability as a lab to perform good data. QC is actually the process where, all right, those experimental samples are in-house, you are ready to run them. QC is the process you build into running those samples to ensure that the data is high quality as it's being produced and that you can check on it after it's been acquired to be able to say, yes, this is good quality data. So QA, making sure you're prepared, that everything is in place to be able to generate good data. QC is, did you generate good quality data? How do you know it? How do you check it? I think that's really the cleanest way to think about it. Rick,
You're on mute, Rick.
Won't be the first time, though. Close.
Okay, I was muted. Sorry. Jennifer, did you have something to add to that, or were you telling me I was muted?
I was telling you you were muted.
Thank you. I'm trying to keep the background noise to the minimum. So thank you very much, Annie, for that. It's a really good set of definitions. And what I should say, I mentioned MQAC earlier, and actually MQAC has a rolling coordinating committee, and we're very lucky to have the 3 members of that coordinating committee on at the moment, who is Annie, Jonathan, and Jennifer. So it's great to have those on board. And Jennifer, I'm going to come to you next. So we talk about quality assurance and quality control. It is extra work on top of our normal day-to-day work. Why should we even bother to apply QA/QC in our untargeted metabolomics work?
Well, I could throw that question back and say, can you afford not to? So I think that there are 2 major reasons to look at your quality management strategy. And quality management is just the generic term that covers quality assurance and quality control. And I think the first reason is you want to have confidence that the data and then the conclusions that you're drawing from that data are robust. And how can you know that your data is robust unless you've got specific measurements to know this? And for your measurements to be of a level where you're confident that they're robust, you need to have had the quality assurance in place to reach that level. So robustness of data and reporting that robustness also has the advantage that other people then also can have confidence in your data. So that's probably the major reason. But there's a second reason, which is a very personal reason, which is, in my experience, having good quality management in a lab massively reduces your sense of personal frustration. It allows you to save potentially time and money as well. But in terms of knowing that what you're running is likely to have a very robust outcome because you followed good quality procedures, you know that you'll spend less time in having to reproduce things, in having to repeat things, in having to reproduce things. And at the end, you go home feeling very confident that what you're reporting is likely to be reproducible in another lab, or at least to the best of your abilities.
Brilliant. Thank you very much, Jennifer. I think just to add to that quickly, we would expect, and we'll talk about reporting standards and how to report quality assurance and quality control later on, but applying it, but also reporting it is really important as well. The third question that we have that we've been asked is as you can see, we're slowly coming into what QA and QC is. So the next question is directed for Ian, and it's what different types of QC samples can be applied in untargeted metabolomics?
Okay, well, that's an interesting question, and one of the things you can ask is, why do you need QC samples at all? And my remarks aim mainly at LC-MS. It's not that you don't need QCs in NMR, but NMR is an inherently stable platform. Whereas many people know my views about the instability of LC-MS. I sometimes describe LC-MS as the work of the devil because it's very, very complicated, because you're trying to go from a liquid to a vacuum in a very short space of time and get things to ionize in a reproducible way. And you're injecting dirt into your mass spectrometer and they don't like it. So the response changes with time. So the question you ask yourself is, what is the QC for? Now, the initial QC that we came up with, and again, this was slightly prior to the HuSaMet project, which I worked on with Rick, but it certainly helped to change our views of how to do it, was what we call the pool QC. And this is the simplest QC that you can have. And it's basically, you have a rack of samples, which you're aliquoting. I can mine the aliquoting even if I can't do it properly. You're aliquoting samples for analysis, and as you aliquot them, you take a little bit of each sample and put it into a pot, and you end up with a pool, especially if you're not very good with aiming the sample. So you have a pooled QC that is effectively a mean sample of all the things you're going to analyze. And the idea is that if you analyze that sample at regular intervals throughout the run, you can look at the responses for all the components in the sample over the whole length of the run. So if your mass spectrometer loses sensitivity, as they often do, you can correct for it. If it gains sensitivity for some analytes, as it sometimes does, you can take that into account. If you see one peak going down, another peak going up at the same time, you know that one is decomposing to form the other. So you get an idea of stability as well. So it helps you to look at the stability of your instrument and of your sample. Now that's fine, very useful if you're doing a couple of hundred samples. Aliquoting a couple of hundred things twice, well, it's never been a problem for me because I've always had one of my students doing it, so it's a very easy task. But if you have many thousands of samples to run from the same study, one of the things you can't easily do is prepare a pool. And in those circumstances, it's often useful to have what Matt Lewis defines recently as a long-term reference standard, but basically a large bulk sample. And again, we use this in the HuSaMet study. We bought some really filthy Sigma bulk plasma and aliquoted lots of it, and used that to study to run with every batch that we ran. And it was also very useful in that it gave us a historical sample as well, so we could compare the run that we'd done on Wednesday with the run we'd done next March, because we had the same QC. So that long-term reference can be used both as an in-run standard and as a long-term standard. these things are also especially useful if you have very limited sample sizes. So if you're working with finger prick samples or something like that, pediatric samples or small animal samples, you don't have huge amounts of sample just to use to not waste, but to use for QC. So there's the pooled sample, there's a long-term reference sample. You could probably, if you were rich enough, use NIST 1950 as a QC if you're doing plasma or serum. Or you make up a bulk sample of your own. And recently we've begun to think about a new type of QC, which is the phenotypic QC. So if you imagine you have two groups, A and B, you can make a pool of that, and that's very useful. But of course, by mixing A with B, you're diluting out some of the stuff that's in B and vice versa. And some of those things that you're diluting out and you might miss in your QC, the pool QC, may be valuable. So it's handy to be able to compare the two groups. It could well be they have different impurities, or there are things in there which change the ion suppression. So there's all sorts of QCs, and it very much depends on what you're trying to do. So if you're trying to look at long-term stability, you clearly need a long-term reference sample. If you're looking at within-run stability, then the pool sample made from the samples you're going to run is ideal. There's no simple answer to this question. You can design, as I hope I made clear, I can see Rick laughing at this. The QC is essential as a minimum for understanding how your instrument is playing on the day. But you may want to use it for other things as well. So you can dilute your QC and get an idea of linearity. You know, are any of your peaks topping out? You know, there are some peaks that show absolutely no statistical variability. That means they're not good peaks, that they're saturating your detector. So you get all sorts of diagnostic information from the QCs as well. And I think that's probably enough.
Ian, can I ask as well, would you classify a blank sample and a system suitability test sample as QC samples as well?
Well, they are designed to ensure the quality of your analysis. So yes, they're sort of QCs, but we do use them differently. I mean, you know, I'm one of those people that worries a great deal about running blank samples in the middle of an analytical run, because I've just conditioned my column, or passivated, whatever term you want to use, and then I inject a blank in the middle and decondition the next 5 samples. So, you know, I mean, I'm one of those people who's beginning to think that you might be able to use your QC sample as your system suitability test. And I'm aware there's all sorts of problems with that. I think what you must do with any QC that you're using is actually think about it. I mean, you know, your brain is the most expensive instrument in the laboratory, and it's usually run on switched off mode. So, you know, what do I want the QC to do? And does the QC that I'm using do that?
No, I would agree. I mean, personally, I think a blank sample is a QC sample. It's just applied in a different way.
Yeah.
For those of you who don't know about SST system suitability test samples, these traditionally, I will say, is just a mixture, a small number of metabolites in a clean organic solvent or solvent, which are injected at the start of a run, a batch, the week, just to check that your analytical instrument is working, and you're basically making sure that acceptance criteria you've set are being met within there as well. So we might come onto that later on. Okay, thank you, Ian. I'm going to move on to Jonathan now, and Ian's probably answered some of this. Sorry, Jonathan, but what can a QC sample be used for? And I spoke of the different types of QC samples, like the intra-study pooled QC, the inter-study pooled QC, blank, etc.
Yeah, no, it's a great question, and I really think this can be considered part 2 to the answer to Ian's question, because, you know, as you ask that question, you cannot think about these 2 issues separately. If you're thinking about the type of QC sample you use, you have to think about how you're using it. And conversely, if you're thinking about how to use a sample, you have to consider the type of QC sample, its history, its composition, its various components, all of that. So what I will try to do to not repeat what's already been said is maybe go into a little bit more detail in some of the areas that Ian already discussed, just to help people think about how they can use their QC samples. So, and Ian, you did a great job of hitting all these different types of materials or types of QC samples. Let me start thinking about it from a QA perspective, from a process perspective. So the first thing you could do with a QC sample is do method development and verification of your method. So this is particularly important when you want to have some idea before you run your precious study samples, how is my method going to respond? You cannot go into that blind. So you could, again, think of a type of QC sample that you would expect to respond in a similar way to your study samples, and use whether that's a reference material that's a matrix matched to your study samples, or if you have a long-term reference sample pool available already, or maybe you even have a pool QC from a similar matrix. Maybe not exactly from your study samples, but a representative subset of samples that could be used for that as well. Once you have a method that you're happy to use, and Ian mentioned that you can use it to condition the system. Now, which one you use obviously depends based on a myriad selection of factors, but some things to think about when using a QC sample for this. You should think about, what does it mean to have your system conditioned? Do you have a predetermined set of criteria that you're trying to meet as you're injecting multiple iterations of that conditioning sample? Or do you have confidence that if you inject a certain number, you're going to hit that no matter what, and then you just do that moving forward? There's obviously different ways to do that. When you're analyzing or your study samples, your data in real time as it's rolling off of the instrument, you can use QC samples to assess variability even within a batch as it's running, particularly if you have select metabolites that you know are present in your sample. You want to know how they're responding in real time. Those can be things that are already there. They can be maybe internal standards that have been added in specifically for that purpose. You also need to think, as your batch, really before you start your batch, how often are you going to inject your QC samples? Because that's important in both within batch assessments, but also when you're done collecting the data and you're going to do a post hoc assessment. So that's kind of the next thing you can do, is assess the variability within your study. And oftentimes the type of sample used for that is referred to as an intra-study QC sample, which of course can be a pool QC sample, it can be something else. And oftentimes this can be done by, maybe once you've processed your data and you can throw everything into a PCA scores plot, sort of all your study samples and your intra-study QCs, and get at least a visual assessment of the variability of your process, whether that's just the instrument or the instrument and the sample preparation, and the variability contained in your samples. Of course, you can also use more quantitative metrics and evaluate like percent RSD for each feature or metabolite across your entire study. And compare that even to just that variability and your interest of a QC sample. So there's a lot that you can do to assess variability, but you can also use QC samples to correct for variability, which particularly in the realm of LC-MS-based metabolomics is hugely important. And of course, for that, you need to consider things like frequency of injection. That'll play heavily into that. The type of sample that you're using for that purpose plays heavily into it. Not only can you assess and correct variability within a study, though you can also assess variability across studies and even potentially correct for those as well. And this can be done between separate studies in the same lab on the same type of sample. So in that context, we think of it as an intra-lab QC sample being used for that purpose. Again, the exact identity composition of that, I believe Ian gave a lot of great details on, so I won't repeat. But you can also use QC samples to compare data between two or more labs, and that's in that context, we refer to that as an inter-lab QC sample. Other things that you can do feature filtering for data set quality improvement. And Ian mentioned a dilution series of QCs, which can be used to assess linearity. I mentioned peak area RSD, which can be used to control for precision in your data. You can also, blanks were mentioned, so blanks are actually hugely important and can be used for background removal of contaminants, things that might confound downstream data analyses that you can control for by using that type of QC sample in your study. Something else I think that was alluded to, but I could just give a little more detail on, is if you are on to evaluate specific features of interest in your data, or conversely, things that you don't want in your data set. So if you have class-specific metabolite changes, you can make sure you're picking those up if you use a phenotypic QC. One thing that we like to do, especially in the environmental and ecological assessment realm, is compare exogenous versus endogenous components in your sample. For instance, if you have an exposure to a particular chemical or even a complex mixture, you can, and if you're mainly interested in the biological responses to that, you want to exclude for exogenous things, particularly those coming from your exposure, or at the very least identify those and pull them out so that you're being transparent on how you're getting the separations between your classes. Lastly, and I'm not going to go into this at all because it's a hugely important area of discussion and probably deserves its own podcast, but you can use QC samples for metabolite identification and annotation as well.
Brilliant. Thank you very much, Jonathan. As Jonathan has said, there's a wide range of different ways you can use the QC sample, both the pooled QC, but not discussed here, the blank samples and the SST samples as well within there. So I'm going to come back to Ian, and the next question we got set, you've partly answered, so I'm going to ask this in a slightly different way. You've said how to prepare a pooled QC sample for a biofluid, say urine or serum, etc. But how do you apply that if you've got a tissue or a cell culture? How would you prepare a pooled QC sample from that, Ian?
That's a really good question because it's actually quite hard. I mean, the first thing you need to do if you're preparing a QC is, okay, let's go back to the University of the Obvious 101. If it's not a liquid, it's a solid or a semi-solid. That means it's very difficult to mix. If it's a lump of liver, or worse, a lump of kidney, which bit do you go for? You need a representative sample to be a QC, ideally. Do you take various lumps of stuff and mix them together and then homogenize them? Or do you homogenize your samples first and then take an aliquot of the homogenate and mix that? And these are all good questions to which we don't have a good answer, if I'm honest. It depends again on pragmatism. I mean, if you're dealing with mouse feces, it's fairly easy to get 50 different mouse fecal pellets into one tube and grind them up. Okay. If you're dealing with human feces, you can imagine the problem is somewhat different. And so again, it's back to this thing, Rick, of what's the sample being used for? And I think it's probably less important that it accurately reflects the mean composition of the samples that you're going to be analyzing, but it is quite important that it is as representative as you can get. So if your samples are small and you can separate out bits of them, I say the mouse feces one is good because you generally get quite a lot of mouse feces and the pellets are small, so you shake up the pellets that you've collected from each individual mouse, and then you whip a couple out, put it into a pot, then you can freeze that knowing that later on, when you unfreeze it, you can do the extraction on it as you would. If it's tissues, say tumor or something like that, it's precious. You probably only got a small piece of biopsy. If it's from kidney, you could probably say, well, actually, I'll use a control kidney rather than a tumor-bearing one. Now, that is obviously not like what you want to be watching, but it does make a bulk QC. And you can do that from the tissue. Now, if you want to make a within-run QC, I would suggest you work on the extract. So you make the extract, then you sub-aliquot it. It's fairly easy to do. And it will at least measure the sort of variability in what you've actually extracted. But try not to kid yourself that you're seeing reality. You're seeing a very edited version of reality. By comparison, urine is dead easy. I mean, you just mix aliquots of urine and they disappear into each other. Plasma is also relatively easy, although there you have to start worrying about what happens when you try to get rid of the proteins. So again, Rick, you're right. It's a simpler biofluid, but if you extract it with methanol or acetonitrile, and then you look at the runs that you get, they will be different. So acetonitrile is not particularly good at extracting the cholesterol type molecules that you see at the end of a run. Methanol's better. Okay, so as soon as you start adding stuff to samples, you're beginning to bias the result. Now, you're doing that with the samples you're analyzing. You're also doing it with the QCs. Trouble is, with a liquid, at least everything's being biased in the same way. So, when you look at plasma, you're not seeing what's in plasma. You're seeing what you've extracted from plasma, and that depends on the solvent that you've used. So, Jonathan's suggestion of using the QCs as a method development tool is a very good one, because you can develop extractions and see which ones gives me the most. But solids are a lot harder. So again, sit there and think about what you're trying to do in the analysis and what you want the QCs to monitor. Now, you can add internal standards to the solid, but if you do that, you have to think very carefully about what you're being told, because you've got a solid sample to which you're adding some liquid, and then you're doing an extraction. Now, 2 seconds thought, if you use that 2 seconds to think, will tell you that you're going to get a very good extraction of your internal standard, and it will tell you absolutely nothing about what you've extracted from your sample. But if you don't have that thought, you will believe that it's telling you something about your extraction. So be very, very careful. The solids are really hard, and I think I've said that enough times now, Rick. I will stop.
Okay, thanks, Ian. I think it's a very important point that you come up. Think about what you're using a QC sample for and how it will be used is really, really important. And do that before you even get into the lab. I think that's absolutely key. Okay, so I'm going to move back to Annie. Because I know Annie has a lot of experience with really large studies as part of her work with Metabolon, or working for Metabolon. And so, Annie, I'm just wondering, we've talked about pooled QC samples, which are, say, for a plasma or urine study, where you've got 100 samples in the study, it's quite easy to combine aliquots of those into a pooled QC sample. But where you've got thousands of samples, and all those samples might not have been collected before the metabolomic part of the study starts, how do you prepare a QC sample that can be used to assess within run or within study variability?
Yeah, so many of this has already been discussed, but it's such a great point. And I think it really is showing how exciting the field of metabolomics is that we're starting to do these longitudinal studies where we're collecting people and then wanting to compare that to samples we collect from those same people in a year, in 2 years, in 3 years. These incredible longitudinal studies, we're seeing a great deal of this work already coming to fruition in previous trials, but moving forward, more of this. And so the mandate from targeted metabolomics is, how do I ensure that I can collect data not only today to tomorrow and compare these, but this year, next year, 5 years from now? The QC samples are how you do this. But just like you said, I don't even have that. They haven't even been collected yet for the samples that I'm going to be acquiring in 5 years from now. How do I start incorporating this QC into today? And we've already mentioned a number of these strategies. The goal of any pooled QC, the goal of any QC is to try to represent the sample type that you are running the most, right? So if it has the most similar makeup of even things like dietary components, you know, inevitably when we see we don't have perfect match populations between that pooled QC sample, the differences don't lie in your core biology metabolites. Glucose is going to be there, leucine's going to be there. Where you start to see the differences is in endogenous or exogenous molecules, things like the dietary markers, the over-the-counter medications. Those may be critical, they may not be so critical to your ultimate interpretation. Regardless of all that, there's just a great deal of success that we've had generally in what we've always already talked about, where just buy a large pool of plasma, serum, whatever you need to get your hand on. We usually use bio-IVT because you can actually choose things like this age group, this sort of breakdown of ethnicity. If you know you are going to be looking at a diabetes trial or something like that, bio-IVT can actually embed some of those disease signatures in so that you can try to match what you ultimately know your population is going to look like. It is not going to be perfect, again, but what we see are the sort of lacks, the limitations of that approach, or again, are mostly in those exogenous markers, dietary, over-the-counter exposures, not your core biology. That type of approach, when you can't create that pool because you haven't even acquired all the samples yet, purchasing, if you can embed some of those disease samples, if you know a priori what the disease signatures you're going to be looking at, we've gone as far to buy 70 liters of a human pooled plasma because we don't want to just bridge one study to another study. We want to be able to bridge all studies to all studies because we think that's going to be of benefit to the entire metabolomics community, that, you know, we can run this same control sample for your cohort from, you know, Copenhagen, from your cohort from Mexico, and there's an underlying unification there. So that is, again, one of the benefits of having this larger reference population to be able to use as a QC sample. But again, maybe not perfect for that study, but a lot of the core biology is really solid with that approach of just purchasing a surrogate similar matrix as you can get.
Brilliant. Thank you very much, Annie. I think there's 2 points that come out there. We're trying to do the best that we can to represent everything the best that we can, but you know, trying to do that extra 5% to get perfection isn't necessarily required as well. So that's great.
Absolutely not. I mean, we've said this already, fit for purpose. Every lab doesn't need to have every quality control sample there is possible, right? It can probably be pretty overwhelming when you think about the list of QC samples that are possible out there. If you're running one study of 50 samples and that's all you need, there are lots of little things that you probably don't need to do to know you still have really good quality data, right? So really it is understanding what is needed to understand the quality of your study. It doesn't need to be so overwhelming.
Brilliant. Thank you very much, Annie. Ian, we're going to come back to you, and one type of aspect that we've not talked about in untargeted metabolomics is internal standards. And you know, many people will think about internal standards being important in targeted assays, but not necessarily in untargeted assays. But internal standards can be included to assess the quality of the data or the quality of extraction and data, etc. within there. But how do you choose an appropriate isotopically labeled internal standard for a specific assay or sample type?
Okay, well, that's again a very tricky question. And if you've managed to get this far without switching your brain on, now is the time when that has to go into overdrive, because again, this is one of those situations where the question, what do you want to use the isotopically labeled internal standard for, is really important. And I suppose where I'd like to start is a warning. In metabolomics, as opposed to lipidomics, which is slightly different, you cannot use 1 or 10 or 20 internal standards to quantify everything that you will come across in the sample. Because mass spectrometry is very irritating in that compounds have to ionize in order to be detected, and different compounds and different structures ionize differently. So spoiler alert, I occasionally reject instantly publications I see where they've used tetrachloroethylene or some such compound to quantify all the things they've seen in their metabolic profile, because they clearly don't, the authors clearly do not understand what they are doing. So we're going to be intelligent and we're not going to be ignorant. We're going to say, okay, I want to use a load of internal standards that are, for example, of different log Ps, so they will appear at different places in the chromatogram. Because what I want to do is very quickly assess retention time stability in a run. So I'll find a range of compounds. I'll use isotopically labeled ones, possibly even probably endogenous ones because they're available relatively cheaply. But I don't want to be covering up a metabolite that may actually be interesting. So if I choose tryptophan as one of my isotopically labeled compounds, and that happens to be one of the biomarkers, boy, have I made a mess for myself. So for them, use an isotope, preferably if you've got a really nice high resolution instrument, a deuterium labeled isotope, because that will tend to elute in reverse phase chromatography before the analyte that it is mimicking. And the deuterium should be spotted very easily, and you'll say, ah, that's not a biomarker. That's an internal standard. And for looking at retention time locking, I think that's a pretty good strategy. Now, one of the problems with isotopes is they are moderately expensive compared to buying standard compounds. So you can choose non-isotopically labeled compounds if there's xenobiotics, like drugs, for example, which also cover a nice range of log Ps, if you know they're not going to be present in your sample. So, for example, laboratory animals should not contain the entire US pharmacopoeia. Cell culture should definitely not contain the US pharmacopoeia. Plants should not contain the US pharmacopoeia. So, but having chosen the compounds, and this works with the isotopes as well, please do a blank run of your matrix, i.e. one that you haven't spiked the internal standards in, and make sure there's nothing in there that looks like them. Okay? Because that's a problem. You assume the internal standards aren't there, or an ion that could be mistaken for them isn't there. And you can be fooled by that. The other really useful thing from I guess this is a QA point of view, is if your internal standards are being aliquoted in by a human as opposed to a robot, they should be present in every sample. Doing every sample to get retention time locking. If you find a run where 10% of the samples don't contain the internal standard, that takes you an awful lot about your procedures. And the other thing that an internal standard can be used for very nicely is checking injection to injection reproducibility. So the internal standards, and I think I'd probably favor the isotopically labeled ones most if you can afford them, but if you can't afford them, I say some xenobiotic or other is perfectly good. But please, 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 in LCMS, don't try to quantify anything with them if it's a metabolomic run. Now, in lipidomics, it's different, but I don't really want to get onto that piece of turf at the moment. And in NMR as well, it is different. Okay, so because you don't have the ion suppression, it's more stable, you can use a single internal standard to quantify if you want. So there you go.
Brilliant. Thank you, Ian. That's a great answer. And again, think before you get in the lab. I think there's a general message coming out today. So I'm going to come back to Jonathan now and ask Jonathan what the community is working on to develop and promote best practices for QA and QC in the metabolomics community.
Okay, so good. This question, I guess, represents a little bit of a shift away from what are the specific things you can do to what are we doing on the whole as a community. And really, in in all of science, but particularly in this regard, we have to recognize, right, that we all stand on the shoulders of giants. And, you know, how far back do you look to start to answer a question like this? Well, you know, I'm not going to go all the way back to the inventors of the mass spec or the NMR, or even as far back as Linus Pauling. What I will try to do is hit the highlights of the last 10 to 15 years, and just state this is not going to be a comprehensive answer. If I do not mention your favorite paper or initiative, and it is by no means intentional. But I would go back to 2005, to the start of the Metabolomics Standards Initiative. I think it's a good starting point for answering this. There were a series of papers that were published by this initiative, I think 7 in total, that detailed minimum reporting standards. And then there was even an 8th one that was like a history of the MSI paper. And that actually would be a great place if you wanted to learn about things that happened before then, because it did a good job summarizing QA/QC focused publications prior to that initiative. Since then, okay, so recognizing that the last MSI paper was published in 2007. So since then, there's been a lot that the community has done, and they can be broadly categorized into papers published by key experts in the field. One that comes up quite a lot in circles that I run in is the Broadhurst et al. Paper that was published in 2018 in the journal Metabolomics. But there are several others, even ones published by people on this podcast. So I know you guys know about them. and these were just that. These are members of the community who are experts in their field saying, hey, we need to think about this. Okay, so there's been that going on. Alongside of that, and you can think of it perhaps as momentum has gained in the field, there have been a number of consortia throughout the years that have sought to address particular aspects of this, okay, and that are currently in varying stages of activity. You know, a couple that come to mind are the Cosmos Consortium of Standards and Metabolomics, whose sort of remit is facilitating integrated data access in metabolomics, which is something that we haven't really even touched on yet, but it is important in this topic. Another would be COMETS, the Consortium of Metabolomic Studies. Annie talked a lot about facilitating large-scale collaborative research. There's a consortium that's done that. One that is particularly relevant in the regulatory toxicology field is the MERIT Consortium, promoting use cases, best practices, reporting standards for metabolomics and regulatory toxicology. Now, I wanted to give a fair assessment, I think, of what's been happening, but I also want to hit on what was mentioned at the beginning, the MQAC Consortium, Metabolomics Quality Assurance and Quality Control Consortium. That's one that's very dear to most of us, if not all of us here on this in this podcast. Just a brief history. This Metabolomics Society gave there was a task group that gave a questionnaire in 2015, which led to an NIH think tank in 2017, which led to the formation of MQAC in 2018. And since then, so over the last, I guess we're going close to 6 years now, I really think that MQAC has gained a tremendous amount of momentum and is currently positioned as one of the leaders in the field to promote the development of best QA/QC practices, as well as dissemination and harmonization of those practices. So I will highlight just a couple things that MQAC is doing. We have over those years, we've done extensive amounts of community engagement, gathering information from the community and collating that information into a cohesive framework of best practices that can be that we are working to put together into a living guidance document for this purpose of to go back to, to develop and promote best practices, right? And so, if you've been at conferences, specifically the Metabolomics Society conferences over the last 4 or 5 years, you will have seen an increased presence of MQAC at those conferences. And so hopefully this isn't the first time you're hearing about it, but I think that MQAC is probably here to stay at this point. And so you can expect to see more good things coming.
Brilliant. Thank you, Jonathan. And I should say that MQAC is a voluntary organisation in a way, and you can become a member. You can apply to become a member if you feel you've got something to contribute. Then please do feel free. So the website is www.mqacc.org. Okay, so we've got about 5-6 minutes left. We've had some really good answers, some detailed answers, which is great. So I'm just going to focus now for the last 5-6 minutes on acceptance criteria. And Annie, I'm going to come to you first and ask you, so if you've got a pooled QC sample that you're using in some way to assess the quality of the data, how do you decide what acceptance criteria you should apply?
Yeah, such a great question. And I think the most important part of this question is understanding that the tighter your QC metrics and the tighter those data pass QC metrics, the finer biological signatures you're going to be able to see. I think a great example is the difference between you being a perfectly healthy human being and you being a raging diabetic is a 20% change in your glucose. 20%, right? In an untargeted methodology, that has to mean you have to have a method that is so precise for glucose that it can accurately see a 20%. So a little bit of how strict your criteria are a little bit dependent on how tight of a biological signature are you hoping to capture. If you are trying to capture as biological shifts as small as something like diabetes, which is obviously incredibly impactful to human health, you have to have very tight criteria. However, there are some diseases, think inborn errors of metabolism, where the actual disease signature is a 1,000-fold increase in the signal intensity of a molecule. You could have an RSD of 100, and you are still going to see accurately that disease signature with no problem, right? So I do think we should always strive for high quality data, right? I think intrinsically as scientists, we want to be able to see the fine, really small biological stories there. But that doesn't have to be where you start. Again, thinking about taking baby steps into this sort of quality control world. A lot of people in the field, if we just look at sort of, again, Jonathan did a great job. The MQAC Society has done a great job reaching out to the community. What are the metrics you use? What I will say is a lot of the community will typically use what is a good QC limit from your pooled samples. So looking at individual metabolites, what is glycine like over your technical replicate? What is its RSD? A lot of people will typically use about 30%. They like to see that variability below 30%. Some of the more clinically based folks are striving for sub 10% RSDs from those technical reproducible measurements, again, on a per analyte basis. There are some folks, like my organization, we actually don't filter any of our data based on RSDs because of the idea, again, that is a biological signature is strong enough to overcome a little bit of noise. We don't want to lose that signature because we've overly restricted it as a result of RSDs. But that's still not to say we don't know exactly what our process variability is for all of those things, because that's really critically important to know if any given molecule is going to be relevant for a disease signature. But again, in terms of what is used in the industry, a lot of people are using 30% RSDs for those, molecules, and QCs, folks in the clinic, 10%. But it really is about what is the disease signature. And of course, the end, just to get even a little bit more detailed, if you don't know a priori, a lot of us don't necessarily know what is the disease strength, a little pilot couple of disease, couple of might get you in the ballpark to know how big of a signature you're going to see. And then you can do some power studies and quickly sort of sort through what are you needing to aim at for your method to be able to get what you need. So there are some strategies to be able to get to, gosh, how good does it need to be to see a disease signature to help you make those decisions?
Brilliant. Thank you very much, Annie. Just for a final question, I'm going to move over to Jennifer and ask, so if you are applying acceptance criteria within your process, what should you do if your acceptance criteria are not met? So do you carry on with the data analysis? Should you reanalyze the data? What are your views on that, Jennifer?
Well, so this is a question which I'm going to borrow a phrase from Ian and say you have to engage your brain. And the first thing you need to do is stop and think, because there's not necessarily a clear answer to this. What you need to do depends on what the aims of your experiment were and whether you can still meet those aims despite the fact that your acceptance criteria have not been met. And we've put acceptance criteria there for a reason. And so one of the first things you need to ask yourself is, why hasn't it been met? And what's gone wrong in the process that I haven't met my acceptance criteria? And there's a huge difference. For instance, let's say I've run a 5-batch study, and at the end of the 5th batch, there was a massive catastrophic failure in the air conditioning. And suddenly it's had all sorts of effects on the mass spec, accuracy. Now my intensity, my peak detection is wrong, and my intensities are now not accurate, and now my QCRSDs are completely out of the ballpark. Well, if I've got a good quality assurance process in place to monitor lab temperature, I should know when the lab temperature was steady, which part of my data is reliable, and then I can look at whether I can just use up to the last QC before the catastrophic failure. I'm still confident that that data is robust. If I take a scenario where the temperature in the lab has been fluctuating over those 5 batches, then suddenly that's a very different story, and I'm less confident that that data is robust, and that may then be the occasion where I start having serious discussions about repeating the experiment. So I think this idea of really thinking about what it means for the robustness of your data is really important before you can answer the question about what you do next.
Brilliant. Thank you very much, Jennifer. We did have other questions, but we're nearly on the hour because we've discussed in such detail, which is good. So maybe we'll run a second podcast on this, and hopefully the panelists would come back to answer further questions on that. But I'm going to finish this off here, and I'm just going to say thank you to all 4 panelists, to Annie, to Jonathan, to Jennifer, and to Ian for using their time today, which isn't just this hour. They have contributed to this beforehand as well in setting the questions and thinking about the topics to be discussed as well. So I'd like to thank everybody for being involved today. For all of the non-panelists that have listened in, this podcast will be available on the Metabolomics Society website in the future and on YouTube as well. That is normally occurs within a month of the podcast. So start looking at the end of February for that. And finally, thank you to the non-panelists who have dialed in today and have listened away to us discussing QA and QC. And hopefully the discussions will continue after the podcast. Thank you very much, everybody.
Thanks a lot.